No mai haere mai, welcome. We are recording. I'm Nick Waipara. I'm here today to start our fantastic Rangahau Rising series, a monthly kōrero with the early career researchers, both Māori Pacific and those in the Bioprotection Aotearoa. So welcome. I'm going to start today with karakia and karakia timatanga for today's hui. Kia hora te marino, kia whakapapa pōnamu, te moana, he huri rahi mā tātou, e te rangi nei, aroha atu, aroha mai, tātou e, huie, tai ki. Kia ora and welcome. So, without further ado, and our inaugural speaker today is Dr. Sarah Inwood. She has many hats. One of the hats is Genomics Aotearoa, but also Bioprotection Aotearoa. She is one of the leaders in future biocontrol, one of the key exciting themes that we have in Bioprotection Aotearoa. And she's going to talk today about a number of things that she's been doing, her research, and I'm going to hand it over to her in a minute when I can see the next page. And today's talk is Rethinking Biocontrol in the Genomics Era. It's the impacts of overlooking the microbiome. So no mai haere mai, welcome, Sarah. It's a great pleasure and honour to have you here today to share your wealth of knowledge, research, and hot off the press from your kōrero last week in Genomics Aotearoa in Auckland. So I'm really looking forward to what you're going to share with us today. So kia ora and welcome.
Kia ora, Nick. I'll just share my slides.
Yep, we can see that.
Perfect.
Excellent.
Tēnā koutou, tēnā koutou, tēnā koutou katoa. Ko Sarah Inwood toku ingoa. And as Nick said, I'm a postdoc for Bioprotection Aotearoa. I'm based at Otago, Whakaihu Waka in Ōtepoti, Dunedin. And I'm going to talk to you today about some genomics research that I've been doing on biocontrol. So biological control, or biocontrol for short, for those that are unfamiliar, is a form of pest control that's used very frequently worldwide. It involves using a natural enemy of a target pest to control it, and that natural enemy is often not native to where it's being used for biocontrol. These enemies can control their pests in a number of different ways. They could be a predator of them, a parasite like a parasitoid wasp, or a pathogen or a competitor, for example. Now, biocontrol has been used for a really long time, and biocontrol success rates have improved remarkably over the course of that time due to advances in the field, including but not limited to increased host specificity testing to prevent off-target impacts and more thorough efficacy testing prior to release to make sure that biocontrol agents do control their target pest. However, the field of biocontrol is increasingly acknowledging that genomics has a lot of power to further improve biocontrol outcomes, but that it's been somewhat underutilized so far. So genomic research is starting to be performed on some biocontrol agents, though more commonly research is performed on species that just share similar life cycles and traits to those organisms used for biocontrol. This research can be focused on understanding traits that are important for the ability of a biocontrol agent to kill its pest. So a couple of examples here are looking at resistance to a pathogen used for biocontrol, or understanding more about how predators locate their prey using olfaction. There's also research being done into resistance to stresses that biocontrol agents are likely to encounter once they're released. So heat stress or insecticide exposure. And there's also a little bit of research being done on traits that impact the environmental safety of using biocontrol agents. So this is an example here where research was done trying to compromise the ability of a biocontrol agent to reproduce so that they couldn't outcompete other native species, but that they were still able to effectively kill their host for use in outbreak scenarios. But a lot of this genomic research and biocontrol has only really gotten off the ground in the past few years, and there's still a lot more scope for genomics to improve biocontrol safety and efficacy. And that's particularly important today, as getting approvals for collection and release of biocontrol agents is much harder now than it used to be in the past. So I'm studying this biocontrol system that targets the Argentine stem weevil, the beetle that you can see on the right-hand side here. I'm using genomics to understand more about the biocontrol of the Argentine stem weevil, which is an invasive pest that was accidentally introduced to Aotearoa from South America in around about the 1920s. And this weevil is a really bad pest here in Aotearoa, as it damages economically important pastures like ryegrass. And biocontrol is one of the primary control methods that we have over the Argentine stem weevil. And that biocontrol uses a parasitoid wasp here, Microtonus hyperode. So this parasitoid wasp is from the same home range as the weevil. It comes from South America, and it attacks and kills the Argentine stem weevil. It actually can't reproduce without the weevil host being present, and that reproduction is all asexual. So all of the wasps of Microtonus hyperode that we have here in Aotearoa are female, and every time they reproduce, they're effectively cloning themselves. For those unfamiliar with parasitoid wasps, this is what their life cycles look like. So in the case of Microtonus hyperode, it hunts its host down in the field and finds an adult weevil and stings it. But unlike when we get stung by a bee in the garden, this wasp not only injects venom into the weevil, but it also injects a wasp egg. And that wasp egg then slowly starts developing and essentially eating the weevil alive from the inside out. It's only 6 weeks later, when the wasp larva erupts out the back of the weevil abdomen, that the weevil is actually killed. That larva then weaves a cocoon, develops into an adult wasp, and the life cycle continues. So Microtonus hyperode was collected from 8 different locations in South America in 1990 by scientists working to establish biocontrol against the Argentine stem weevil. It was collected from 8 different locations that you can see on this map here in order to increase the genetic diversity that was available for these biocontrol releases, because as I said, this wasp reproduces asexually. These populations of wasps were then brought back to Aotearoa and mass reared in containment over an 8-year period between 1991 and 1998. So each of those 8 populations was kept separate, and all but one of those 8 populations was made up of multiple clonal lines of wasp descended from single individuals. Throughout this rearing and containment, there were staggered releases of Microtonus hyperode throughout Aotearoa. Every time a release was made by the scientists working on this biocontrol program, they released equal numbers of each of those 8 populations, again trying to maximize the genetic diversity available in these wasp populations. They did that so that if certain populations were better suited to, say, establishing in the top of the North Island versus the bottom of the South Island, that they would be able to. So part of the reason that we're studying this biocontrol system is through some of our colleagues at AgResearch, we have access to an incredibly valuable collection of samples. While this mass rearing and containment was going on, the scientists doing this rearing and this research were constantly freezing samples from these populations from different clonal lines at lots of different time points during this rearing program. And then when they were making releases in different locations in Aotearoa, they were then going back to these locations 6 months, a year, 2 years later, and collecting samples of wasp to see what had actually established at those locations. And having access to these samples allows us to do some really cool genomic research to better understand factors that might be influencing biocontrol efficacy. What I'm going to focus on today in my talk, though, is research that we've done into the microbiome of Microtonus hyperode. So research on eukaryotic biocontrol agents tends to ignore that when you release them, so for example, when Microtonus hyperode was released throughout Aotearoa, their bacteria and viruses and fungi that they carry were also being released into Aotearoa. And research outside of the scope of biocontrol seems to indicate that different components of the microbiome could definitely be influencing traits that are important for efficacy as a biocontrol agent. So for example, parasitoid wasps like Microtonus hyperoides, which are very commonly used as biocontrol agents against insect pests, actually often carry viruses. These viruses are generally beneficial, although sometimes detrimental to their ability to parasitize their host. And so by overlooking the microbiome of these biocontrol agents, we're overlooking something that there is much potential to be influencing their efficacy. And so I started off looking at the virome of Microtonus hyperoides using both DNA and RNA sequencing. And in doing so, we found a novel double-stranded DNA virus in the wasp that hadn't ever been found before. We named this virus Microtonus hyperoides filamentous virus, but throughout the rest of this talk, I'll just refer to it as MHFV. It's a bit easier to say. So what we have on this slide here is a phylogenetic tree of double-stranded DNA insect viruses that infect insects. We've got MHFV sitting here in this yellow box down here. So MHFV sits in this viral family here, Filamentaviridae. It's a new viral family. It didn't have a name until last year. And again, until last year, the only other virus that had a complete genome in this family was this LBFV, Leptospirina billardi filamentous virus, which again infects a parasitic wasp. Most of what we know about double-stranded DNA viruses in insects is restricted to Baculoviridae and Nudiviridae viruses, because these viruses are fairly often used as biocontrol agents for other insect pests. But we know remarkably little about Filamentaviridae. This is highlighted by the fact that almost half the genes in the genome of MHFV, we have no idea what they do. They have no significant similarity to anything in the databases we compared them to. But we do know a little bit about LBFV. It has quite an interesting impact on the host wasp that it infects. LBFV manipulates the behavior of the parasitoid wasp it infects. Normally, uninfected solitary parasitoid wasps only parasitize a host if they've never been parasitized before, if they don't have another wasp egg in them. And so when you look at the frequency of the number of eggs inside of a host, you'll see a big peak at 1. You'll have some hosts that haven't been parasitized yet, but you'll very rarely see multiple eggs inside a single host. So Microtonus hyperoides is also a solitary wasp like this. For solitary wasps, in most cases, the first egg in the host develops successfully into an adult wasp, and if there are ever any more eggs put into that host, what's called superparasitism, those younger eggs are competing with a more developed egg or larva, and they very rarely survive. However, when Leptospirina billardi are infected with LBFV, suddenly they superparasitize their host like crazy. You can see here that they were detecting hosts of Leptospirina billardi with as many as 8, 9, 10, 11 eggs inside of them. This superparasitism behavior that's induced by this virus actually helps to spread that virus through the population of wasps. And it actually results in lower overall parasitism rates by Leptospirina billardi because they're wasting their eggs in hosts that have already been parasitized. And that results in these infected wasps being outcompeted by other uninfected species. So this superparasitism behavior is maladaptive or detrimental to the wasp, but this virus does provide some advantage to the wasp as well. It helps to protect the host immune system from mounting an immune response to the wasp egg when they are parasitized. And that increases the rate of success in parasitism in the absence of that superparasitism behavior. So a related virus to MHFV is having both beneficial and detrimental effects on the parasitoid wasp that it infects. It seems the scope that MHFV could potentially also be influencing traits that are important for Microtonus hyperoides as a biocontrol agent. So when we discovered this virus, we found it in contemporary wasp samples that had been collected from Aotearoa in the last 5 years or so. But having access to this incredible sample collection through our collaborators at AgResearch, we were really interested to know how many of those populations they collected from South America carried MHFE. Were all of the wasps they collected infected with it? Was it restricted to certain populations? And so with the help of Jacob Grupp, who was an honours student I co-supervised last year, we used qPCR to test every single clonal line of wasp from each of those 8 collection locations in South America, early and late in the rearing process, to detect which populations were infected with MHFE. Now, it's important to note we don't have access to the samples that were taken from South America or very, very early in that rearing process. They were destroyed, but we do have access to samples from still fairly early on in rearing. So what we're looking at here is a bar graph showing the infection prevalence of MHFE, where we're down at 0. That's none of the wasps that we tested in a population carry MHFE. Up at 1, every single wasp we tested in that population carried the virus. So we're looking at the proportion of wasps that had MHFE early in rearing. This is 1992 to 1993. So the data is somewhat sparse here because some of these lines had very few samples frozen early in rearing. And this is also still preliminary data. We'll try to improve our sample numbers in future. And that means that these 95% confidence intervals are quite wide on some of these bars in this bar graph here. But most importantly, the populations that were collected from Chile don't have any virus detected in them early in rearing. Now, these populations in Chile are separated from the rest of these populations that have varied prevalence of MHFE by the Andes mountain range, which is quite a significant barrier for movement of wasps back and forth. Things look a little different, though, when we look at the same virus prevalence later in rearing. Now, every single population that we've tested has evidence of this virus, and every population, except for one from Chile, has 100% prevalence of this virus. Again, we've still got pretty wide confidence intervals, and we'll try to improve our sample numbers where possible in the future. But this is a really dramatic change from early in rearing to late in rearing. So what we think has happened is that in some stage in this mass rearing process that was done over 8 years in containment, it wasn't known that this virus existed. And so we think this virus has spread from what were originally infected populations into what were originally uninfected populations. This is something called viral spillover. How can we confirm that this is viral spillover during rearing? How can we get more confidence in this? We can have a look at the variation in the genome of MHFE between these different populations. We would expect clonal lines from the same population to have much more similar viral genomes to each other than they would to other populations. And if we do see that early in rearing, but then that pattern is messed up and looks kind of random later in rearing, that gives us much more confidence that we've had virus moving from one population to another, rather than just levels of virus that were so low we couldn't detect them. So what we're looking at here is a principal component analysis that is based on just over 3,000 single nucleotide polymorphisms in the genome of MHFE. In this graph, points that are closer together are much more similar to each other, and points that are far apart are much more dissimilar. And we've coloured all of these points here based on the population from South America that these samples are derived from. So again, we're looking at these samples from 1992 and 1993, 2 or 3 years into this rearing process. And we see, for the most part, pretty clear geographic structuring of variation here. What I mean by that is that all of the samples from Mendoza cluster really closely together. This is the population that only had one wasp collected in South America and therefore one clonal line established from it. When we look at another population collected from Argentina, Hilario escasubi over here, again, these samples all cluster in the same corner of the PCA, although we've got a few different groups here. This is a result of there having been multiple wasps collected from this location in South America, and therefore multiple clonal lines of this wasp in that mass rearing process. So we do have that geographic structure we expected early in rearing. Do we still see it later in rearing, or do things look messed up? And where do the Chilean samples sit? This is the same PCA, but with samples from 1994 and 1998 added on top of the graph. And so if we have a look at where these Chilean samples are, they're in pink and purple, they seem randomly scattered all across the PCA. We've got purple points from conception in the top left, top right, and bottom right. And we've got samples from La Serena, the other Chilean sampling location, down the bottom and up the top. And so what this likely means is that these samples here caught virus from this Mendoza population. These samples here caught virus from that Hilario escasubi population, for example. And so this supports that spillover idea of MHFE moving from infected into uninfected populations. It also looks like we've had spillover between populations that were already infected too, though. If we have a look early in rearing, all of the samples from Uruguay clustered down the bottom of the PCA here, but later in rearing, there's a big cluster of them up here. And so again, that helps to support this idea of spillover, not only between infected and uninfected populations, but also between infected populations. And this really helps to highlight the risk of ignoring biocontrol agent microbiomes. When this rearing was being done, it wasn't known that this virus existed. And even today, when biocontrol releases are made using eukaryotic biocontrol agents, we typically overlook the fact that they have a microbiome, that they have bacteria and viruses and fungi inside of them. But this finding really helps to highlight the risk of overlooking that. We don't know if, for example, Chilean samples without virus would have been better biocontrol agents. But the fact that we also have access to samples collected from the field after release helps us get a bit of an idea about that. So we can start to also investigate prevalence of MHFE in these samples that were collected after those biocontrol releases in Aotearoa to again see how many samples did or didn't carry this virus. And things look quite interesting here as well. We're looking at the same graph here as we had earlier on, but this time looking at field collections rather than samples and rearing. We're looking at infection prevalence, 0 being no samples infected, 1 being all samples infected. This time we've separated samples out based on the year they were collected from the field. And our 95% confidence intervals are still here. They're not quite as wide as they were early in rearing. The main takeaway from this result here is that every single post-release sample that we've tested so far has been positive for MHFE. Every single wasp collected from the field carried this virus. And that's really interesting because we have samples from as early as 1992 and 1993. And we know that this early in that mass rearing process, we had a couple of populations of wasp that were uninfected. And every time releases of Microtonus hyperoideae were being made, they included equal proportions of those populations. So we should have had uninfected wasps being released into the environment. But then when we go back to these collection locations early on, we don't detect any trace of uninfected wasps. So did these uninfected wasps from Chile not establish in Aotearoa once they were released? Or did they establish, but they were infected by this virus after they were released? We can tell the difference between these two options here by looking at variation in the wasp genome this time instead of the virus genome. If we can tell the difference between eastern and western wasps relative to the Andes or Chilean wasps from all of our other populations, we can tell if none of those western Chile populations established, or if they did, but they just have virus now. And so again, we're looking at results from a principal component analysis, but this time this is based on SNPs in the mitochondrial genome of the wasp. And when we look at variation in the wasp mitochondrial genome, we see really nicely that most of our Western samples from Chile separate out on the PCA, as well as some samples from San Carlos de Bariloche in Argentina. This location here is actually in the foothills of the Andes. And so we can discriminate between Eastern and Western wasps. And these Western wasps early in rearing shouldn't have had virus. So if we add samples that were collected from the field after they were released onto the top of this PCA, what we're looking for is how many samples do we see sitting down here? We should expect them to be roughly the same proportion as they were made in releases if there was no difference in the establishment between Eastern and Western wasps. So this is the same PCA. This time all of those rearing samples are in yellow, and in this teal color here are samples of Microtonus hyperoides that were collected from the field after those biocontrol releases. What we see is that only 9 out of the 94 samples collected after release, so roughly 10% of the wasps collected after release, clustered down the bottom here with these Chilean populations. So we do see a trace of these Chilean populations in the field. They're just all infected with virus, but they're also less than the 25% proportion than we would expect them to be. So were MHFV negative Chilean wasps potentially less competitive than those already infected Eastern populations? And the only Chilean wasps that managed to hang on and establish successfully in the field were ones infected with this virus? The researchers that were establishing this biocontrol program at the time actually already noticed as early as 1997 this difference in the establishment between these Eastern and Western populations. They were doing morphometric analyses because they didn't have the funds to allow for whole genome sequencing like we do now. And they were looking at things like wing vein patterns in their rearing samples to see if they could find traits that they could discriminate between those Eastern and Western wasps. They'd then go out to the field, collect wasps, and then see whether the proportions that they classified as being Eastern or Western matched what they knew they had released at those sites. And they saw much higher than expected proportions of Eastern wasps and much lower than expected proportions of Western wasps. These horizontal black lines are what the proportions should have been based on the proportions of those wasps and their releases. And so again, could this have been that uninfected Western wasps weren't as successful at parasitizing weevils in the field? Is this difference in establishment potentially their viral infection status early in rearing? Or were there other differences between Eastern and Western wasps, and this difference in establishment is unrelated to MHFV? We don't know that yet. That's really hard to know when we don't know what impacts MHFV actually has on Microtonus hyperoides. And that's a really hard thing to work out when all of the live wasps we can get our hands on have the virus. We don't have an uninfected control group to compare to. But we do have some data that suggests that it might be impacting some pretty important traits in Microtonus hyperoides as a biocontrol agent. This heat map here is showing some gene expression data in weevils. So every row across this heat map is a different gene in the MHFV genome. We're only looking at a handful of them here. And every column down this heat map is a different weevil sample. So these weevils are grouped together based on whether they were parasitized or not, which I detected using a PCR test. And what we see is that every single parasitized weevil that we sequenced has pretty high expression of a number of genes in the MHFV genome. So it looks like when the wasp parasitizes the weevil, not only does it transmit venom and an egg, it also transmits this virus. We know that viruses carried by parasitic wasps often influence parasitism success. There are plenty of examples, not only LBFV, that show really important roles in suppressing the host immune system to prevent wasp eggs being attacked and killed. So maybe MHFE is being expressed in parasitized weevils because it's playing a similar role here. We know that MHFE can't be required for parasitism of Argentine stem weevils because in their home range, there were uninfected wasps west of the Andes that were successfully parasitizing Argentine stem weevils. But maybe uninfected Western wasps had their eggs killed by weevil immune systems more commonly and didn't establish as successfully. We don't know right now. We need to do a lot more work to find out whether or not MHFE has impacted the efficacy of Microtonus hyperoides as a biocontrol agent in such a way. The other interesting result that we saw here with this experiment is that we actually see in a really small number of our weevil samples expression of virus in unparasitized weevils. And most of these weevils were actually ones where we had observed the wasp attempting to parasitize them, but it had been interrupted. They weren't able to successfully transmit a wasp egg into them. And we're quite interested in seeing this viral expression in unparasitized weevils. This is because of, again, some research done by the scientists at AgResearch who established this biocontrol system. They noticed that weevils that were exposed to Microtonus hyperoides but not parasitized by them, had higher rates of mortality. This graph here is showing the cumulative mortality of Argentine stem weevils on the y-axis over 22 days on the x-axis. And these lines are grouped based on whether weevils were not ever exposed to the wasp, were parasitized by the wasp, or were exposed to them but not parasitized. And there's a massive jump in mortality of weevils that were exposed but not parasitized. So we wonder whether this mortality here could possibly be the result of MHFE being transmitted to weevils that aren't parasitized. This is because there's a similar phenomenon seen with another parasitoid wasp, Aotearoa japonica, where when its parasitism of its host is interrupted, that causes mortality of the host. And that mortality is being caused by the transmission of viral particles. So we plan to test this in future by isolating MHFE from Microtonus hyperoides and injecting it into unparasitized weevils to see if it causes mortality. If it does, then we'll be considering whether we could manipulate this viral infection somehow to enhance the effectiveness of Argentine stem weevil biocontrol. So hopefully through the work that I've shown you today, you can see that continuing to overlook the microbiomes of eukaryotic biocontrol agents is probably having unintended consequences, like this viral spillover we saw in the mass rearing of Microtonus hyperoides. And these consequences could potentially be impacting biocontrol efficacy, stability, or from what we've seen in some other parasitoid wasps, maybe even host range of biocontrol agents. And so now that genomics is a bit more accessible and less prohibitively expensive than it was in the 1990s, we believe that where it's possible, profiling microbiomes of biocontrol agents could enhance biocontrol safety, predictability, and success. In the 1990s, when this biocontrol system was being established, it would have been far too expensive to be doing the sorts of work that we're doing today. But we can learn lessons from what happened, having access to that valuable sample collection, and hopefully improve biocontrol outcomes in future. So this is not the work of me alone. I'd like to acknowledge everyone in the Lab for Evolution and Development that supported this work, particularly Jacob, who's done a lot of that qPCR with me, Kimberly, who does a lot of work on the wasp genomics, and Peter Dearden, our PI down here. Also, a massive thank you to our collaborators at AgResearch, particularly Stephen Goldson and Mark McNeil, who have allowed us access to their sample collections that they had the foresight to keep in the 1990s. It's allowing for some really exciting research to be done. And this work is funded by Bioprotection Aotearoa and a Marsden grant, as well as support from Genomics Aotearoa and the University of Otago. Thank you very much. I'm happy to take any partai.
Kia ora, Sarah, that was absolutely phenomenal, and reminding everyone, oh, homai te paki, you can hear mine, but everyone can give you a virtual clap. And remember, he pātai tā koutou. If you have questions, just put them in the chat. I have one. One's more around the policy that EPA, the Environmental Protection Agency, who, for example, oversees the importation of classical biocontrol insects, are they aware, do they now ask for a risk? Do you know if they do a risk profile of the, say, virome or the microbiome of insects being asked, being applied to be brought into Aotearoa?
I'm honestly not too sure myself. There would potentially be others out there that would be much more equipped to answer that question. But I think it's something that is starting to be acknowledged more in the field of biocontrol. Most of the research that we've done so far on the virus hasn't actually been published, but hopefully when we do publish that, that idea will be picked up even further.
Yes, I mean, they'll be very interested in your publications, I'm sure.
Yeah.
There was, I recall there was a weed biocontrol program on Tradescantia fluminensis, um, overseen by Manaaki Whenua Landcare Research, but funded by the Department of Conservation. And there was the Neolema beetles being brought in to have to as a control, the three of them, and there's a gregarine parasite found in the gut. Now,
Mm,
I mean that was an example of something where that had looked and that did have an impact on the health of these beetles, but
mm.
I'm unaware of your mahi shows that there should have been a wider actually a screening process to see what the microbiome is, further than say an obvious parasite. So I guess what I'm saying is tautoko to the mahi you're doing, and given, do you think it should be going forward that all new organisms, or do you think what you've done with the ag research study also, is it useful to go back and screen some of the major biocontrol agents?
Yeah, I think where funding allows, as always, going back and studying more of these established biocontrol systems has a lot of value in understanding biocontrol a bit better. And where we're bringing in biocontrol samples, I think it could be very beneficial to be sequencing samples to get genomes of what's being released, to understand what viruses or bacteria, for example, are being carried by these biocontrol agents. Because the more we understand about these biocontrol agents before we release them, the better we can safely use them.
Absolutely. So we've got some great partai in the chat. So the first one is, thanks Hester, kia ora. Has signs of the virus been detected in any native New Zealand weevil species?
We haven't honestly looked yet. There was a fair bit of research done by Barbara Barrett and other colleagues at Ag Research before Microtonus hyperoides was released to make sure that it didn't have very large off-target impacts, particularly on native weevils in Aotearoa. But it would be really interesting to go back and check whether any weevils outside of Argentine stem weevil have traces of this virus, because if we do, that's absolutely another reason that we should be better understanding the microbiome of these biocontrol agents before release. If it is being transmitted to native weevils, it could be having detrimental impacts there. But yeah, as of yet, we've only ever looked in Microtonus hyperoides and a few related parasitoids to it and Argentine stem weevils.
Kia ora. Kia ora, Claudia. I agree. This is awesome work and a great presentation. Fantastic. Did you check to see if you find the virus in the home range? Is there a possibility that the virus was already in the weevil in New Zealand before the wasp were released and then got transmitted during the rearing? So I suppose it's going back to, acquiring.
Yeah. Kia ora, that's a really good question as well. Unfortunately, we have floated the idea many a time of trying to get samples from South America of Microtonus hyperoides or Argentine stem weevils today. And logistically, that's a whole lot harder now than it was 30 odd years ago. We would love to go and see if samples in South America still have this virus, which populations do or don't, and how prevalent is it. Um, We do have access to some of the weevils that were collected in South America. They're in our -80 freezer at the moment. And so I absolutely plan to test them in the future and see whether the weevils had significant prevalence of this virus in them as well. We can't rule out yet that the virus wasn't already in Aotearoa before the wasp was released. Again, we have some samples of weevils that were collected from the field before the release of Microtonus hyperode, but a lot of those have been stored in such a way that they're pretty tricky to get DNA out of. So fingers crossed in the future we'll be able to have a bit more confidence about those.
Kia ora, Arnold. Yes, kia ora, Sarah. Thank you for this great presentation. Do you think that in present times where the disease investigations are mandatory before releasing an invertebrate for containment, you would have tried another approach, such as removing the virus out of the rearing colony?
Yeah, I think that's a really great point. But without having known at the time that only some of these wasps carried the virus, maybe we could have released only uninfected populations, or it would have been easier to have done experiments at that stage to understand a bit more about what impacts this virus had. If you took some Chilean samples that weren't infected and then took some Chilean samples and infected them, that would have been a really great way to understand that maybe a lot of what drives the efficacy of this biocontrol system is actually the virus. And so maybe releasing wasps without the virus would have made them much worse biocontrol agents. It's hard to know, but as a part of a Marsden that I got at the end of last year, we're hoping to be able to use RNAi or antiviral treatments to try to get rid of the viral infection in some of these wasps so that we can start investigating some of those aspects a bit more.
Yes, the virome is one of those unexplored universes, isn't it? That and you're deep, you're going, yeah, that exploration you're doing is going into places and spaces of the virome. Who knows? Kia ora, Craig. Nice presentation. Microtonus hyperide shares its host Argentine stem weevil with Microtonus aethiopoides. Oh, sorry, I hope that's okay from a pathologist's pronunciation, particularly the iris strain. Do you think the virus could spill over to another Microtonus species such as Aotearoa, and also New Zealand has 3 native Microtonus species? Kia ora, Craig. Thanks for that.
Yeah, it's a great partai as well. We have looked in the genomes of the Microtonus aethiopoides strains that are used for biocontrol in Aotearoa, and we haven't seen any signs of MHFE in them, though that hasn't been nearly as an extensive investigation as what we've done specifically in Microtonus hyperode. And it's really hard to know, but I think that there is certainly potential that the virus could have spilled over. We do see virus sometimes in weevils that aren't parasitized. And as Craig says, we do see still frequently in the collections that we get from around Aotearoa some Microtonus aethiopoides parasitizing Argentine stem weevils. So if they were to parasitize a weevil that carried the virus, it seems very possible that this virus could spill over into those aethiopoides strains, but we haven't seen it yet.
Oh, ka ora miharo. Ruby has answered my question.
Oh, perfect.
So she thanks you for your wonderful, brilliant presentation. Yes, the EPA does conduct a risk assessment when considering the application for releasing biocontrol agents. The decision-making framework involves weighing up environmental, economic, societal, human health, and cultural values. So she's given a link there to the EPA website. So kia ora, Ruby, that's awesome. And kia ora, Amy. So interesting. Do you have plans to investigate whether viral efficacy against weevils to cause mortality may be impacted by interactions with other microbial members of the microbiome, say bacteria and fungi?
That would certainly be an interesting thing to investigate. We've done a bit of 16S sequencing of Argentine stem weevils, and in weevils infected with or not infected with MHFE, we don't see huge differences in their microbiome. But that's certainly, we haven't investigated that premature mortality yet. We just plan to do it in future. And so if we start to do those experiments, then absolutely we could be saving samples to follow up with 16S and see if other microbes, be that fungi or bacteria, are influencing those interactions.
Kia ora for that, Pātai. Kia ora, Phil. What do you think the mechanism for viral spillover? How does it occur? And his comment there, I guess in the field it could be through one wasp strain successfully parasitizing a weevil that had been unsuccessfully parasitized by a virus infected strain previously. So all that interactions going on there.
Yeah. We honestly aren't sure at this stage. We plan to have a bit more of an in-depth discussion with our collaborators at AgResearch, Stephen and Mark, who are intimately involved in this mass rearing, to get our heads around that a bit more. But having not been there when the mass rearing was being done, we are honestly not sure. The fact that we didn't know that this virus existed until a few years ago means that the mass rearing, while it was designed to keep those different populations of wasps separate, may not have been sterile enough when sterilizing equipment used on the same populations or between populations, for example. But yeah, not sure right now.
Sure. Ngā mihi hanarea, wonderful Rangahau Sarah. Are there any other viruses that are also transmitted? Also, are there any viral variants and would they be of future concern? Good point.
From RNA sequencing, Microtonus hyperoideae alongside the DNA sequencing that we've done, we have picked up traces of other viruses that they carry, just through the nature of them being RNA viruses rather than DNA viruses. They're a little harder to follow in a lot of the work that we've done because we have been largely just using DNA sequencing on these wasp populations, in part for my work on MHFE, but largely actually for Kimberly Dainty's work on the wasp itself. It just so happens that when we sequence a wasp, we also sequence this virus. But that's definitely something that I would be interested to look into a bit more in the future. MHFE is the only virus that we saw that was related to another virus that had a demonstrated role in parasitism, but that certainly doesn't mean that it's the only virus being transmitted during parasitism. We don't know much about the variation in the genome of this virus right now, and we're certainly interested to dig into it a whole lot more. Not only does it look like the wasps that are established in Aotearoa all have this virus, they all seem to have particular viral strains. There doesn't seem to be much representation of the virus that was in the Mendoza samples from Argentina to start off with. But again, without knowing what this virus does, it's really hard to speculate on whether different variants of this virus might be better or worse for biocontrol or more or less dangerous.
Yes, kia ora. Kia ora hanarea. I also just have to comment on the fantastic I guess having that whole collection from Craig and others at AgResearch acknowledging that keeping those samples and curating those for future studies has just been, I mean, it's outstanding that that's happened. So, kia ora.
Yeah, it was incredible to have the insight to see that genomics could have power, even though it wasn't accessible at the time. And yeah, the fact that Stephen and Mark and those at AgResearch that did this mass rearing kept those samples, but have then allowed us access to those samples has been huge in terms of the research that we can do.
Yes, kia ora. Kia ora, Saba. Fantastically long question. So I'm just going to, great, mahi, Sarah. To enhance biological controlled strategies against weevils, is it worth investigating whether plants emit those volatile organic compounds when attacked by the weevils and whether these VOCs can attract the parasitic wasps?
Yeah, that's something that we do plan to actually do in the next round of funding that we've got through Bioprotection Aotearoa, both study this virus, but also investigate olfaction, how Microtonus hyperoideae detects its host. Is it relying on volatiles from the weevils, from the plant? There's been a little bit of work in this space, but nothing that we can really directly answer those kinds of questions on. And as Sapar points out, it's been shown in other systems that pallidna viruses carried by wasps can sometimes influence those olfactory detection. So absolutely, that would be something interesting to look forward to in the future too.
Kia ora. And Saba also has added in around the checking of female and male wasps for the presence of the virus, particularly given female wasps harbour these viruses in the ovaries. So that's
Mm.
just a comment from Saba. Kia ora for that.
Yeah, so all of these wasps that we're sampling are female. Only female wasps were released into Aotearoa. But when the ag-research scientists were getting these collections from South America, they did find a handful of impotent male wasps. So that's this asexual reproduction of the wasp is again something that we would like to understand a whole lot more, and whether the virus is different between male and female wasps, if we were ever able to get access to male wasps, would be very interesting. We've had a look at where this virus is in different tissues in Microtonus hyperrhodae. I was fully expecting it to largely be in the ovaries, but it seems to be in other tissues much more than it is in the ovaries, which was really interesting.
Kia ora. And ngā mihi, Stephen Goldson, has a comment around the virus. We read a million, obviously, cycling containers, etc., may have also harboured the virus. So I guess that's just a comment. But also acknowledging the whakapapa or the history of the system right from the Bioprotection Research Centre days, which Stephen and others have led. So it's sort of fantastic to see that research continue into the future that you're leading, Sarah, and your team, and others in your team in Genomics, Aotearoa, and Mihi to Peter Dearden and the rest of your team, but also seeing, that history of people like Barbara Barrett and Stephen Goldson and others that have been doing this mahi for years.
Yeah,
And
we couldn't do what we're doing now without their work in the past.
And it's just fantastic to see that, people that think research is you reach an end. Well, look what the opening of the whole new technologies that wasn't possible to do, like exploring the virome and the microbiome through, the genetic technologies now that you have there, just
Yeah.
very powerful. So kia ora. And just fantastic partai, everyone. Just wonderful. So we've sort of reached the end, but I hope you've seen just a little sliver. You know how to get hold of Sarah personally if you need to continue this kōrero. But honestly, just wanted to reiterate our thanks for being the inaugural kōrero today, Sarah. Just wonderful, and all the best for your research into the future. So kia ora. Speaking of Hanaraya, we have her kōrero in a month's time, so August the 20th. So look on the Bioprotection Aotearoa webpage and you can register, look at the series. You can see the links to Sarah's kōrero and also a recent article of her mahi, but also to see other links that you'll see from the webinar series. Also acknowledging the huge mahi of Bioprotection Aotearoa, Zoha, Anna, kia ora, thank you, Amanda and others. The team there doing amazing work, and so are you all. So just wanted to reiterate our thanks, Sarah, for my pakipaki for your kōrero today. Any other questions, just put them in the chat, and I will close our hui today with me te karakia whakamutunga. Any last words, Sarah, just before I do?
No, that's all from me. Thank you very much.
Kia ora. Okay. Me te karakia whakamutunga. Pouhihiri, pourama, pou te whakaaro, pou te tangata, pou te aroha, te pou e hara nei e te tātou, te hei Māori ora. So take care, everyone, and we'll see you next time. Ka kite anō.